In the PDB file or the MSIF file for a protein or peptide, what you'll see is that each atom is going to be given a unique identifier. And it looks a little like gibberish, but it's really just showing you the element followed by its position in reference to the alpha carbon. So if it's the alpha carbon, it's going to get an A, but then if you're one away, well, now you're in the beta position, so it's a B. 2 away, you're in the gamma, so you are G. 3 away, delta, D, etc., etc. So here's just an example with arginine and lysine. And you can see too that once you get out here, you're at this NH1 and NH2. So this is for the one in the first branch and the second branch. But the H is not indicating the hydrogens there. Remember, the second position is indicating the location. It's really easy to think, oh, the NH1, NH2, those are indicating the hydrogens on this nitrogen. No, they're actually indicating the H is indicating that it's 6 away from the alpha carbon. And the hydrogens, if you wanted to list the hydrogens would start with an H, but then they'd still be given that same H positioning because they're given the number of the atom which they're attached to. But often you're not even going to see the hydrogens are going to be listed because many files actually don't have the hydrogens incorporated. But if they did, they would have an H. In this case, everything in this structure was just the protein, and so they're all going to be labeled as atom. Your main things are going to be labeled as atoms, so your proteins, your nucleic acids. If, however, you have any sort of ligands or you have solvent in there, those are going to get this het atom indicator. And that het atom indicator is telling you it's part of a hetero or different molecule than kind of the standard ones like your proteins or nucleic acids. In this case, we have these small molecule binders, these ligands, and then you'll also see water. And these heteroatoms are going to be listed after your main atoms. Speaking of the main atoms, you might see this OXT. OXT is indicating this is the terminal oxygen on that protein, so this is your C terminus.